Assalamu alaikum, dear listeners. The topic for the today's discussion is restriction enzymes. Restriction enzyme is a particular group of enzymes that recognize a specific sequence in the DNA strand and then make a precise cut. The action of the restriction enzyme produces restriction fragments that can be further used for the DNA fingerprinting. Restriction enzymes are also used for the isolation of the gene of choice from the genome. Restriction enzymes were for the first time discovered in 1970 by Hamilton Smith, and he was later in 1978 awarded a Nobel Prize. These enzymes are naturally produced inside the bacterial cell where they cut the foreign DNA and eliminate the chances of the infections. The bacterial cell genome is protected by the action of the restriction enzymes by the methylation of the adenine and cytosine nucleotides. There are a variety of the restriction enzymes, each recognizing a specific sequence. The restriction enzymes type 2 that make a specific cut are important in the field of molecular biology and biotechnology. Restriction enzymes are named according to the bacteria from which they are derived. The first three letters in the name of the restriction enzyme represent the genus and the species name and are written in italic. The fourth letter represents the bacterial strain from which the enzyme is isolated, and the Roman numerals indicate the order of the discovery. The specific sequence that are recognized by the restriction enzymes are called as the recognition sites. Recognition sequences are usually 4 to 6 base pairs in length, and usually these sequences or sites are palindromic in nature. The fragments produced by the action of the restriction enzymes are called as the restriction fragments. These fragments are useful in determining the DNA fingerprint. The restriction fragments produced by the restriction enzymes have two types of the ends: blunt ends and the sticky ends. When a fragment do not have a single-stranded terminals, the ends are called as blunt ends, and when the single-stranded terminals are present, the ends are called as sticky ends. Sticky ends are of two types: 3 overhangs and 5 overhangs, depending upon the terminal carbon of the deoxyribose sugar. If the 5 carbon is on the terminal side, the overhangs are called as the 5 overhangs, and if the 3 carbon 3 OH end is on the terminal, then the overhangs are called as the 3 overhangs. There are a variety of the enzymes that bind the same recognition site. Such enzymes are called as the NEOSCHIZOMERS. Although these NEOSCHIZOMERs bind to the same recognition site, but they cleave the DNA in between the two different nucleotides.